Hello everybody, my name is Faramarz Farihi. I am going to give you a talk on brain-inspired spiking neural networks for neuromorphic deep learning and neuromorphic reinforcement learning methods. In this talk, we consider insects' olfactory system as a feed-forward neural network, then similarity between basic structure and function of insects' olfactory system and rodent hippocampus. Then we consider some papers on bio-inspired spiking neural networks and their application in machine learning and cognitive robotics. Then I present my proposed research. In order to implement cognitive capabilities of animal brains in artificial systems, researchers have used mainly neural networks information. However, recent progresses in the field have demonstrated the critical role of cellular and molecular information for developing novel artificial systems. There are some challenges for this research field. The first challenge is modeling by simplification of experimental data at different levels of information. The second challenge is implementing the algorithm regarding computational complexity point of view. Insect's olfactory system is actually a well-known feed-forward neural network. Others in environment trigger olfactory receptor neurons in the antenna to a spike that is transferred into antennal lobe and mushroom body. It is interesting to note that in this architecture there is a convergence of information that happens between antennal lobe and antenna, while divergence of information takes place between antennal lobe and mushroom body. Eventually, output neurons may be triggered to elicit behavior. In this neural system, we know biophysical properties of olfactory receptors, the number of neurons in antennal lobe and mushroom body. We have good information on electrophysiology data on different neurons of the system and behavioral data. Interestingly, there are similarities between structure and function of insect's olfactory system and rodent hippocampus when we consider the number of neurons, the connectivity, and electrophysiology features of the neurons in these systems. In rodent hippocampus, sensory input is transferred into entorhinal cortex, then to dentate gyrus, and in insect olfactory system, information from antenna is transferred to antennal lobe, then to mushroom body. The first interesting fact is that neuronal population ratio in EC to DG is about 1/6 in mice, and neuronal population ratio in antennal lobe to mushroom body is about 1/5 in flies. In addition, in both mushroom body and dentate gyrus, excited neurons show sparse spiking, low firing rate, and in both mushroom body and dentate gyrus, neuronal population show sparse coding. It means that the number of neurons activated by a stimulus is low. In both mushroom body and dentate gyrus, inhibitory neurons play critical role in pattern separation efficiency. Now we take a look at some works on bio-inspired spiking neural networks and their application in machine learning and cognitive robotics. The structure and function of antennal lobe and olfactory receptor neurons in antenna and mushroom body neurons, that is called canyon cells, and output neurons have been used in developing classifiers. This work is a spiky neural network model inspired by other discrimination in honeybee's olfactory pathway as a deep learning architecture that works as a probabilistic classifier trained in a supervised manner. Novel feature of this method is the role of lateral inhibition in the information processing and also in data classification that increases the performance of a classifier on high-dimensional data in a machine learning context. This happens by enhancing linear separability of stimulus patterns by increasing contrast between input dimensions. The method has been used to construct a spiky neural network for the classification of multivariate data. In this work, real-value multivariate data are converted into spike trains using virtual receptors, and their output is processed by lateral inhibition and derives a winner-take-all circuit that supports supervised learning. This work shows that neuromorphic network achieves classification of generic multi-dimensional data. In a similar work, electronic nose sensor data has been classified by a insect olfactory system inspired spiky neural network. In this work, classification was achieved in real time using a GPU accelerated spiky neural network simulator. I think this work can be used for developing navigating robots with electronic nodes. We have recently constructed a spiky neural network as a 3 layers feed-forward neural network: sensory layer, hidden layer, and output layer. Hidden layer includes 2 sets of neurons, excitatory neurons and inhibitory neurons. Biological backpropagation takes place in output neurons, and backpropagation induces production of retrograde messenger into inhibitory neurons. Synaptic plasticity between inhibitory neurons and output neurons is affected by retrograde messenger. To measure pattern separation efficiency of this system, pairs of stimuli with overlap in their activation pattern were presented to the system, and pattern separation efficiency was measured in output neurons as the value of overlapping between activated neurons in output neuron layer. The model's parameters were set such that output neuron shows sparse spiking, low firing rate, indifferent excitation to inhibition ratio such that the system can adapt itself to imbalance E to I ratio. This happens in a short time. This happens by changing synaptic plasticity between output neurons and inhibitory neurons induced by backpropagation. The simulations show high pattern separation efficiency of the system in different levels of excitation over inhibition ratio, while the system without such backpropagation and retrograde messenger mechanism cannot be robust to excitation over inhibition ratio fluctuation. We have realized that the network size has a remarkable impact on algorithm complexity to be implemented. Comparing time for an iteration of algorithm shows that Hippocampus model works faster than leaky integrate fire neuron model for this spiky neural system. We have constructed a spiky neural network inspired by basic structure and function of the Drosophila olfactory system that we named in silico fly. In this spiky neural network, simplified hypothetical retrograde messenger production and diffusion from output neuron into mushroom body cells has been modeled. To our knowledge, this is the first work that implements second-order conditioning in a spiky neural network. In these two-dimensional simulations, in silico fly navigate the environment by random walking, and after a few pairing of odor and electric shock, the in silico fly avoid the odor, the blue one. When trained odor, blue one, and null odor, green one, are paired for a few times, in silico fly may avoid the second odor, here the green one too. Okay, now after a short introduction, I present my proposed research. Neuromorphic engineering aims to reproduce brain computation using VLSI technology. Some applications are vision artificial systems, auditory artificial systems, and microelectronic nodes. A CDP learning rule modulated by reinforcement signal is one of the most interesting research in this field. To implement such methods in neuromorphic hardwares, some aspects should be considered. The input type and the level of complexity that is intended to be implemented. The first research field that I am interested in is neuromorphic deep learning using spiky neural networks. Deep learning has been used in simulation of some perception tasks, and one of the algorithms used in deep learning is backpropagation algorithm, and to apply this type of deep learning in neuromorphic technology. It is interesting that classical deep learning architecture is not exactly neurophysiologically plausible. Therefore, novel neurophysiological deep learning methods may be a great progress in the field. Some challenges and research topics are the storage of weights as the main bottleneck for hardware implementation of deep learning, the training time of deep learning. So efficient online algorithm for deep learning on hardware is a novel topic. spiking hardware implementation of deep learning, and neuromorphic sensors combined with deep learning neural networks, and finally hardware models of biology inspired learning, such as learning with cortical circuits. Regarding the research topics and challenges in neuromorphic deep learning technology, I propose developing neuromorphic brain-inspired deep learning methods for data classification using information on cortex, hippocampus, and insects' olfactory system, and artificial olfactory systems that show memory and learning capabilities. And also, I propose neuromorphic deep learning using backpropagation algorithm in neuromorphic hardwares to overcome challenges. The second research field of my interest is neuromorphic reinforcement learning using spiking neural networks. Reinforcement learning is a learning paradigm that is based on learning by interacting with the environment. Developing reinforcement learning in a spiking neural network context is a big challenge. Moreover, classical reinforcement learning and dopamine modulated synaptic plasticity are not analogous. STDP rules have been implemented in neuromorphic hardwares like Spinnaker, but it doesn't explain learning that is based on feedback from environment. The future researches that proposed by Mikaitis 2018 are to implement a reinforcement learning agent like a robot to perform conditioning tasks using the Spinnaker implementation of dopamine modulated STDP, and using a Spinnaker to simulate the spiking neural network based controller in a real time, so it could be evaluated, embodied in a real robot. Regarding this information and my experiences and interests, I would like to work on neuromorphic brain-inspired reinforcement learning using information on insects' brain to develop cognitive autonomous robots with olfactory system that shows high memory capacity and associative learning. And neuromorphic brain-inspired reinforcement learning using information on mammalian brain to develop robots at system level with focus on basal ganglia and cortex. Thank you for your attention.